Hey, hello and welcome back again to another video tutorial of Roy's Academy. In this video tutorial, I'm going to discuss another important aspect of the nitrogen fixation. Okay, in one of the other videos, I have discussed about the nitrogen fixation of bacteria in non-symbiotic method. Okay, and in this video, I'm going to mainly deal with how the symbiotic mode of operation for the nitrogen fixation happens in case of leguminous bacteria. Okay, the nitrogen fixing bacteria. As I've mentioned before also in that video, and in this video I'm also recapping about the brief introduction regarding those bacteria, like the leguminous bacteria or the diazotrophs. Diazotrophs are the bacteria which helps in or which are able to fix the atmospheric nitrogen or reduce it to amino acid or ammonia and supply it to the plant body. And symbiosis is nothing but the mutual understanding or the mutual benefit of two different species as well as two different genus also, a completely different type of organism which are mutually benefited from each other by the supply of certain food materials. Okay. Now in the non-symbiotic mode, the pathway was different. There was two different pathways like I've discussed in that video, and in the symbiotic mode of fixation, it's little bit larger. Okay, it's little bit complex. Okay. In the symbiotic mode of nitrogen fixation, those diazotrophs like the Frankia, Klebsiella, Azobacter, Rhizobium, then Azotobacter, Azorhizobia, Cynorhizobia. Okay. All this kind of nitrogen fixing or legume fixing bacteria, then Leguminosaria, okay, then several other Azotobacter. These are some kind of also examples of the bacteria which helps in the nitrogen fixation. They are also known as fertility bacteria or fertilizer bacteria, okay, which helps in improving the soil fertility by fixing the nitrogen. And this bacteria have a specific type of gene in their body which is known as the NOT factor. Okay, the NOT factor is nothing but lipocetopolysaccharide. Okay, let me draw the NOT factor, then it will be easier to understand. This is how the diagram of the NOT factor is. The board shaped diagram of the NOT factor, it is about the chair conformation, the chair conformation of the NOT factor. This is what it looks like. This is what it looks like. Okay. Roughly a lipocetopolysaccharide, lipoceto-oligopolysaccharide, LCO. Okay. Oligosaccharide. Saccharide is the full form. Now, in this case, the NOT factor is actually this lipoceto-oligosaccharide, which is having an acetyl group along with a chitin backbone that's present in the body of the that nitrogen fixing bacteria like Rhizobium and all those species and all. Now what happens in this case? Let me depict through a diagram. This is actually the chemical structure. I didn't draw the entire chemical structure because it would be huge structure and I cannot explain the entire phenomenon. So I've drawn a rough diagram of the chair conformation, chair and table like anomalic ring chain structure of chair conformation of the carbohydrate molecules. As we know, there is chair-like conformation of the anomalic rings of the carbohydrate. So this is that form of the lipoceto-oligosaccharide. Now let me draw three columns first. Okay, let me erase this thing. I will draw it later. In order to save space, I am erasing it right now. I will draw it later. Okay. Now, let me draw a few columns. It would be easier to depict them. The first column, in the first column, let me write the bacteria. Okay. Now, in the second column, let me write soil. And in the third column, let me write plant. Okay. So this is what the column is. Now, you see the structure right here. This structure is nothing but a microscopic histological, so microscopic structure of the root tip of the plant body. This is present beneath the soil. This particular layer is the soil layer, and this particular portion is present beneath the soil. Okay. Now you see these portions right here present. These are nothing but these portions, these hair-like outgrowths, and these hair-like outgrowths are known as the root hairs. Let me draw a portion of the root hair here. Then it would be easier to understand. Okay, this is the portion of the root hair. Now, in this case, what happens? This is the cellular portion. These are the cells. This portion is nothing but mainly the cortical cells. Okay. Always remember this section of the cell division in the roots are basically starting from this tip of the root. This area, this particular area, is called the zone of cell division. Zone of cell division. Okay, the cortical, epidermal, endodermal cell division. From this particular area, you see that the cells have become little bit larger. Okay, through this entire area, the cells have become little bit larger. This particular area is zone of cell elongation, and this final area is the zone of cell maturation. Now these three sections have different functions. Now in each of this zone, the division of the cells are kind of like the cortex, then the epidermis, endodermis, pericycle, xylem, phloem, and all those things. Okay, from outward to inward, from this outward area to the inward area, if I proceed, then first I will get the cortex portion. Okay, cortex along with the epidermis. After that, the endodermis would be present, and beyond endodermis there would be the pericycle, and beyond pericycle there would be the zone of the xylem, phloem. Now cortical cells will be present here, okay, at the outermost area, and the endodermis will be present inside, okay, and along with the cortex the epidermis would be present, okay. So this particular area of cell is nothing but this one of these areas. Here, let me draw the root hair. One root hair I am drawing here, okay. This is a root hair. This is a root hair, and this portion is the epidermal portion or outermost portion, along with the cortex, the cortical cell division. Now actually with the cortex, the epidermal portion of the cells are present, and beyond the epidermis is the endodermis is present. Now this particular area, the cortex is the only area from where the root hairs begin their projection. Okay, Now, as I said that the cortex is present along with the epidermis. The cortical cell division is, the cortical portion is nothing but the zone of cells where the outermost outgrowths or the cells are present, the epidermal portions are present, beyond that the endodermis is present. Okay. Now, in this case of the plant cells, along with the cortex, the root hairs are present. Now, here what happens, let me jump into the column right now. Firstly, the bacteria, let me draw the bacteria also. Let these be the bacteria here. Okay. Let these be the bacteria, the rhizobium bacteria, which are present in the soil. Okay. This area is nothing but the soil. Okay. And let me put the heading as symbiotic into fixation. Okay. So what happens in this case? Bacteria, the rhizobium bacteria, in the first step, what happens? How the symbiotic nitrogen, there is a big process, huge process actually. In the first step, the bacteria secretes NOD A protein. The NOD gene somewhat looks like this. Okay. This is a NOD gene actually. Now the NOD D NOD D is actually also a protein. And the NOD ABC are present in this direction. NOD A, NOD B and NOD C. These are present in the opposite directions to that of the NOD D gene. Now what happens is, in this case when bacteria secretes, activates the NOD D gene which produces the NOD D protein. Okay. The NOD D protein, this entire thing gets into the soil and this NOD D protein gets activated. Okay, the NOD D protein, it comes into the soil and gets activated. How does it activate? The plant body also secretes a phytochemical or a secondary functional compound of the plant body which is known as the flavonoid. Flavonoid, okay. Now this flavonoid comes to the soil and when it comes in contact with the NOD D protein, it will activate the NOD D protein. Okay. Let me show the activation in the second step, then it will be easy to understand. Okay. So the flavonoid is coming in contact. Now the NOD D protein, NOD D protein is activated. Now this activated NOD D protein will again move to the bacteria. Okay. It will again move to bacteria in the second step, and what it will do? It will target the NOD ABC gene. NOD ABC gene. Now when it will target the node ABC gene, this node ABC gene will get activated. In the third step, the node ABC gene will get activated, and once the node ABC gene is getting activated, it will produce transcription, it will activate the transcription factors and transcribe the RNAs, which will produce proteins. Those are nothing but node A, B, C protein. Okay. Node A, B, C. Now this node ABC protein is activated. The node ABC protein is activated, which is obviously because node ABC gene gets activated and produces the protein. It's already active. It gets activated again with the contact of this flavonoid. With the contact of this flavonoid. In the first step, the node D protein was getting activated. In the second step, in contact with flavonoid. And here, the node ABC activated gene, which produces node ABC protein, is getting activated again with the contact of this flavonoid. Okay, those are secretive phytochemicals or functional compounds secreted by the plant body. Now, the flavonoid, which activates the node ABC protein, this node ABC protein will now again turn back to the bacteria and modify the node factor. Okay, this node ABC protein will again come back to the bacteria and modify. In the fourth step, it will modify the node factor. Node factor modification. Node factor modification. Okay, now this modification of node factor, let me show you how the modification of node factor happens. In the first step of the node factor modification, the node ABC protein is actually a complex of enzyme. Okay, first node C enzyme will add N-acetyl glycosamine, N-acetyl glucosamine to, this will add N-acetyl glucosamine to chitin backbone of LCO, lipocheto oligosaccharide, that is the node factor. And in the second step, node B is a deacetylase enzyme. Node B is a deacetylase, which will remove acetyl group from LCO. And in the third case what will happen? The node A is an acetyl transferase. Acyl transferase. It will transfer fatty acid to the deacetylated portion of node factor, that is LCO. This is how the entire node factor gets modified. Now this modified node factor, what it will do? The modified node factor will start the cortical cell division of the primordium. So the cell, it will look like this. In the second step, this is the first step. In the second step, let me draw. This one is the first one. The second one, let me draw here. The cortical cell division, it will start. That is, here the cortex is present along with the root hair. Okay. Now here the cortical cells, cortex are present along with the epidermal portions. Okay. Okay, these are present, and in this area particularly, the primordium is nothing but the epidermal, the portion where the epidermis is ending. Okay, the epidermis where it's ending or coming in contact with the soil, that is the primordium. Here it will start the cell division. Okay, this is how the cell division will happen. Now as soon as the cell division starts, it will, in the third step, what will happen? This cell division will help in coiling of the node factor, coiling of the root hair, coiling of the root hair. Like this, the root hairs will start coiling. Okay. In the outer epidermal portion, not in the endodermis. This everything is happening in the cortical and primordial region of the epidermal portion of the plant root hairs, or cortical region of the plant root hairs, and it will, this is how the cells have already divided and started coiling. Okay. Now it will develop, along with the coiling, it will develop an infection thread. This is what the infection thread will look like. Okay, this red colored thread area. Now through this infection thread, what will happen? This rhizobium bacteria, which is present in close contact with the root hairs, will start entering through the root hair cells into the primordial area or the cortical zone of the cell division. Okay. Now once the rhizobium is entering here in this particular area, in the fourth step, what will happen? It will form, the coiling will persist and continue, and it will form a nodule-like appearance. Okay. It will form this nodule-like appearance within this epidermal portion. Okay, within this epidermal portion, it will form a nodule-like appearance through which this bacteria, rhizobium bacteria, will form bacteroid cyst. It will form bacteroid cysts within this nodule. Okay. Now, after everything is, after all of this happens, okay, after everything and all of this happens, this bacteroid cysts of the bacteria will start producing will start producing certain enzymes. So let me jump on, start producing certain chemicals, okay, a reduced form of the atmospheric nitrogen. So let me jump on to the column now. So after the knot factor modification, this knot factor will again go to the plant body, okay. In the fourth step, this, let me draw here only, that this knot factor will again go to the plant body and it will form the nodule. Like I showed you the process, the nodule formation will happen along with bacteroid cysts. This nodule formation will happen along with bacteroid cysts like I described in these 4 steps. The primordial cells over the epidermal region of the cortex will divide, and this along with the cortical cell division, cortex will divide along with the root hairs on the outer layer of the outer section of the root hairs. And when the cortical cell division is completed, this area is also known as primordium. This primordium means the basal area or the area which is present in the layer of the epidermal portion or the cortical portion which is present in contact with soil, okay. That particular area is known as the primordium, okay. Now this primordial cell division, when it's getting completed, it will help in coiling of the root hair. So once the root hair gets coiled and it is finally going on coiling and coiling, finally it will form this nodule-like appearance along with the primordial cells which is overlaying over the epidermal layer or epidermal skin which is in contact with the soil. So this nodule formation, this will also help certain cell divisions in this cortical region also, okay, the layer closer to the soil. Since the epidermal layer is dividing, the epidermis, then the cortex is present along with the epidermis and the endodermis is present inside, inwards here to the middle. So the cortical cell division will also happen, which is basically happening in the primordium area, or primordial cells are dividing. Now what happens in this case, as soon as the bacteroid cysts are formed, this bacteroid, rhizobine bacteroid, will fix the atmospheric nitrogen and start producing in the 5th step or in the final step. Let me write down here. In the 5th step or the final step, the nodules along with the bacteroid will start producing certain amino acids like glutamine, okay, glutamine, asparagine, then certain urates or uric acid like substances or nitrogenous end products like allantoin. All these glutamine, asparagine, allantoin and all the nitrogenous nitrogen reduced substances are supplied to the plant body, okay. All these things are supplied to the plant body, and the plant body in turn supplies glucose to the bacteroid cysts for their development or cellular respiration. And the plant body, by taking this end product of the nitrogen reduction, the end products of all these things like all these amino acids and all, the plant body will carry out certain metabolic aspects, cell development and all physiological regulations and all these things will go on inside it. So this is how the NOT factor is getting activated via the NOT-D and ABC gene and their proteins, and once the NOT factor gets modified, the modified NOT factor will go on this successive steps of the primordial cell division, which is present over the epidermal area, okay, that is the outer layer of the outer portion of the cortex. And in the primordium region, the cell division will happen, which will also lead the cell division of the cortical region inside, and that will lead to the root hair coiling. The root hair will start coiling, okay. And once the root hair gets totally coiled, it will form a nodule-like outgrowth. this outgrowth is having the primordial cells divided, a overlayer of primordial cells divided, along with entrapped bacteroid cysts which have entered through the infection thread that was formed during the onstart of the root hair coiling. Now this entrapped bacteroid cysts will produce this all these reduced portions of the reduced end products of the nitrogen reduction or the nitrogen assimilation through certain genes like the heteromeric molybdenum iron and homodimeric iron containing complex, okay, the nitrogenous complex, okay, the MOF complex and all, the NIF genes and all those things which help in producing this enzymatic complexes, catalytic complexes, which helps in fixing the nitrogen along with the hydrogen into ammonia and then converting it to these amino acids, and these amino acids are being supplied to the plant body for the development, and the plant body in turn is benefiting the bacteria by supplying the simple sugars or glucose or plant carbohydrates. So this is how the symbiotic method of the nitrogen fixation happens with the activation of the lipocheto polysaccharide, which is a chitin N-acetyl glucosamine with chitin backbone that is present in the body of the bacteria. Okay, body of the bacteria, specifically in the cell wall of the bacteria, it is present, and it will modify this lipocheto polysaccharide. It is getting modified with the help of these enzymatic reactions, which is thereby coming in contact with the root hair cells and again modifying the root hair cells for the nodule formation, entrapping the bacteroids, thereby starting the process of nitrogen fixation or assimilation and having a mutual benefit, that is symbiosis among each other. Okay. So if you like this video, share the video as much as possible, subscribe my channel, hit the like button and comment down in the comment section below. Thank you.